That consists of an OD sensor, so we know how much feed we're pumping into the muscle tanks. That's one pump. Then we also have in the muscle tank a mesh, so the muscles are not in the bottom, and then we can take all the waste that will accumulate in this corner and with the siphon move it back to the algae tank. And then we have another temperature sensor here in the muscle tank with a pump, so we can regulate the temperature of the muscle tank and cool it down a bit. Here you can see our final setup, how it looks in real life without the blueprint. And here we have the muscle tank, and then in the cardboard we have the algae tank.
Temperature was calibrated and implemented by adding realistic amounts of warm water to the system and changing proportional, integral, and derivative parameters in our muscle UI controller.
The characterization of our pumps. We did this by varying the duty cycle of our pumps and then looking at the flow rate. We did this by varying the duty cycle between 40, 60, 80, and 100%, doing 3 measurements with each and then taking the average of this to increase precision. And then the time measured was the time for half a liter of water to flow from one bucket into another. We then converted this measurement into liters per minute, again using the same variations of duty cycle, giving us a total flow rate that we could use for further calculation.
You can view our server on the enclosed URL with a given username and password. This is where we keep displaying our data, and the data is stored with the Adafruit.io. This is also where you can export it. When it comes to the OD sensors, we made OD sensors work. We measured the values with it, like intensity, OD. We plotted the graph and shown the results.
Top left, we have the filtration function, which is really important for all these following graphs. On the bottom left, we have sort of a flow chart of our DIA process for the system. And on the right, we have a 24-hour model that shows the algae concentration and the muscle concentration being constant where we need it be.
A practical mathematical model. The difference between these two is on the left we don't have a stepper, which means that it's over 1 hour and everything happens instantaneously. The algae is also theoretical, while on the right we have where we should input our sensor data for the algae and a stepper, which means that it happens over time.
Validation of the experiment and the setup, starting with the experimental design of the algae growth rate, the quantitative determination, and then moving on to the experimental design for the muscle feeding and the quantitative determination of the muscle feeding, and then going on to the biocompatibility testing. Here you can see a visual graph of the different flasks for the algae growth rate, which were left overnight and then measured using the apparatus in the system, and likewise for the muscles, which were put into 3 different buckets and data collected after. We start with the algae growth rate, which we prepared in different vials at the same concentration. We controlled the contamination, stored it under natural sunlight, incubated it over 24 hours, and measured the final cell concentration using the instrument as the initial dilution. Then we also conducted growth rate calculations and replications for statistical reliability. Here you can see the graph of the algae growth rate using the initial and final cell concentrations measured over a 24-hour period and replicated 3 times. Furthermore, we also conducted statistical analysis of algae growth rate. Here we have the experimental design and setup for the muscle feedings, which were put into 3 different buckets, and then were fed, sampled, and collected the data from, and conducted filtration rate calculations. Here you can see the graph summarizing all of the algae concentrations over time. And then we conducted biocompatibility on 2 different materials, and we found that both of them were okay to be used. However, the latex one is much lower than the initial and the plastic because the surface area is greater than what we actually have in our experimental setup. Furthermore, we also conducted experimental data on ammonia, which can be seen here.
As you've seen in our setup, we reduced the number of pumps to 2 for reduced power draw. And for our materials, we use mostly scrap material, so that is scrap wood and scrap flamingo. We also use the scrap flamingo for thermal isolation method, where we measured the efficiency. So without the styrofoam or flamingo, it dropped around 12 degrees, whereas with the flamingo, it only dropped around 4 degrees in our bucket over a 30-minute interval. This leaves an efficiency of around 66%.